Dear students, I, Dr. C. Anita, will detail you about chromium. This episode will focus on the distribution, food sources, deficiency and toxicity of chromium. At the outset, let us get introduced to chromium. Introduction. Chromium, an ultra-trace mineral, is found in the body in small amount and are measured in microgram. It is a metal with the ubiquitous presence in air, water and soil, exists in several oxidation states. Biological importance of chromium was first proposed in 1954. However, it was accepted as an essential nutrient in 1977. Chromium is widely distributed in the food, especially unrefined foods and whole grains. It is termed by researchers as biologically active chromium or glucose tolerance factors. Objectives. After studying this episode, you will be able to know the distribution of chromium in the body, enumerate the functions of chromium in the body, understand the absorption and utilization, recognize the food sources of chromium, describe the deficiency and toxicity of chromium. Now, we shall learn about the distribution of chromium in the body. Distribution. There has been relatively little data on chromium content in different body tissues and fluids until recently for adequate analysis of chromium in biological material. The human body contains less than 4 to 6 milligram chromium. Tissues especially high in chromium includes kidney, spleen, liver, lungs, heart and skeletal muscle. Tissue chromium concentration appears to decline with age. Chromium concentration in blood. The blood concentrations of chromium reported in literature was understood with the gradual improvement of instrumentation ranging between 1 and 40 microgram per liter, as reported by Whelan and Peterson 1999. Earlier, Christensen et al. In 1993 has reported that the concentrations are 0.035 to 0.04 microgram and 0.120 to 0.34 microgram per liter for the blood serum of a healthy population and full blood respectively. Chromium concentration in tissues. The total amount of chromium in the human body ranges between 0.4 to 0.6 milligram. Du Bois and Belleville in 1991 claimed that the chromium reserve relative to the body weight is higher in newborn children compared with adults. Anderson et al. In 1997 confirmed the hypothesis. Trivalent chromium tends to accumulate in epidermal tissues, that is hair, etc., and in bones, liver, kidney, spleen, lungs and large intestine. A very small amount of chromium was detected in the brain. It would be a good idea to go through the biological role of chromium as it would supplement the other topics. Biological role. The primary role of chromium appears to be in the maintenance of normal glucose tolerance. It acts as a cofactor for insulin by enhancing the activity. Chromium potentiates insulin action and thus influences carbohydrate, lipid and protein metabolism. It maintains the structure of RNA molecules. It has a possible role in regulating lipoprotein metabolism. Chromium supplementation in normal and hypolipidemic individuals decreases plasma triglycerides and cholesterol and increases HDL cholesterol. Students, let us understand the absorption and utilization of chromium. Absorption and utilization. Chromium present in diets in the form of inorganic compounds or organic complexes may be released from food in acidic solutions in the stomach. It is absorbed throughout the small intestine, especially in the jejunum. Although it is thought to be absorbed by positive diffusion or by a carrier-mediated transporter, about 0.4 to 2.5% of chromium intake is absorbed into the intestinal cells to be utilized by the body. Organic chromium is readily absorbed and quickly passes through the body without being utilized. Inorganic chromium compounds are poorly absorbed. Probably it is 1 to 3% or less, regardless of the dose or dietary chromium status. Hexavalent chromium compounds dissolve and are absorbed better than trivalent chromium compounds when supplemented directly to the intestine, as documented by isotope experiments. The site of or the mechanism of chromium absorption is not clearly known. Some papers give evidence of passive diffusion. Anderson and Kozlowski in 1985, Bunker et al. In 1984 had reported that the percentage of chromium absorbed from diet decreases until it reaches 40 microgram per day, after which the absorption stabilizes at 0.5%. Absorption of inorganic trivalent chromium is indirectly proportional to dietary content. It has been claimed that dietary factors such as oxalate, iron, and high dietary intakes of simple carbohydrates influence the absorption of chromium. The type of dietary carbohydrate consumed modifies absorption from chromium chloride to starch, increases the absorption. Absorbed hexavalent chromium readily passes through the red cells and becomes bound to the globulin fraction of the hemoglobin. Trivalent chromium cannot pass this membrane. It combines with the B globulin of the plasma and is transported to the tissues bound to transferrin. Inorganic chromium is excreted primarily in the urine, with some small amounts lost in hair, sweat, and bile. Human serum chromium levels have been detected to be 1.6 to 3.1 microgram per ml. Hair chromium level is a useful determinant of chromium status. Factors influencing chromium absorption within the stomach: amino acids such as phenylalanine, methionine, and histidine, high carbohydrate, oxalate, and aspirin levels in the diet improve absorption. Amino acids act as a ligands to improve absorption, such as silicilations typically help chromium to remain in soluble form and prevent olation. Vitamin C may also enhance chromium absorption. Lipophilic compounds like picolinate enhance chromium absorption through cells' lipid membranes. Inorganic chromium in a neutral or alkaline environment forms olation compound of high molecular weight, which further precipitates and thus reduces the absorption. Phytic acid present in grains and legumes diminishes its absorption. Antacids reduce chromium concentrations in blood and tissues. Transport. The inorganic trivalent chromium binds with transferrin and is transported along with iron bound to transferrin. Absorbed chromium circulates in blood bound to the beta globulin, plasma fraction, and is transported to tissues bound to transferrin or other complexes at the physiological concentration. Kandor in 1999 and Vincent in 2000 explained that transferrin receptors are insulin sensitive. An increase of this hormone in blood stimulates the transport of transferrin receptors from the vesicles inside the cells to the plasmic membrane. Receptors on the cell surface bind chromium saturated transferrin, which is partly subject to endocytosis accompanied by chromium release at the acidic pH of the newly formed vesicles. Chromium released from the multiple transferrin molecules is sequestered by apochromodylin to produce chromium-loaded chromodylin. Chromium from blood is relatively quickly absorbed by bones, accumulating also in the spleen, liver, and kidney. Storage. The body stores 4 to 6 milligram chromium, especially in kidney, liver, muscles, spleen, heart, pancreas, and blood. Tissue chromium concentration has shown to decline with age. It is assumed that chromium is transported in tissues with ferric iron because of its transport by transferrin. Excretion. Borel and Anderson, 1984, claimed that the average amount of chromium excreted in human urine is 0.22 microgram per day, when the average daily intake being 6.2 to 8.5 microgram per day, which is consistent with the relatively low absorption rate. Ducrose in 1992 explained that absorbed chromium is excreted primarily in urine by glomerular filtration or bound to a low molecular organic transporter. A small amount is nevertheless eliminated in hair, perspiration, and bile. Muhammad Shah et al. In 1998 reported that the minimum excretion of chromium is through milk, as a human study has shown, in which the supplemented isotope 53 chromium was detected in blood but not in milk. Chromium excretion, especially by the urinary system, may increase 10 to 300 times in stressful situation due to a diet rich in simple sugars in contrast to complex carbohydrates. The different factors impacting on the urinary excretion of chromium in humans have been studied by a number of authors. Now we will identify the food sources of chromium. Dietary sources. Chromium is widely distributed in the food and exists in the trivalent form, especially in unrefined foods and whole grains. It exists in foods in complexes with other compounds that make it easily controlled and utilized by the body. The major dietary sources of chromium include meat, fish, poultry, whole grain, and beef. Seafoods like oysters, liver, peanut, and certain dairy products like cheese contain high levels of chromium. Moderate sources of chromium are eggs, butter, margarine, and potato. The chromium content of many fruits like banana, orange, grapes, apple, and vegetables like green beans, spinach, raw mushrooms, spices like cinnamon, cloves, bay leaves, turmeric, tea are good source. Milk and pork are quite low in chromium content. Chromium is generally lost in refining and processing of certain foods. Molasses and brown sugar are rich in chromium than white sugar. Stainless steel cookware may increase the amount of chromium in food. Let us start with deficiency. In humans, chromium deficiency is reported to cause impaired glucose tolerance, weight loss, and peripheral neuropathy. In animals, an increase in mortality, incidence of atherosclerosis, hypercholesterolemia, hyperglycemia, and altered glucose hemostasis are observed. Study of chromium deficiency are relatively scarce. However, Anderson in 1994 has summed up the results of number of trials on humans, rats, mice, and other animal species in a review of physiological and biochemical symptoms of chromium deficiency as weight loss, weak bones, poor skin health, low concentration and poor memory, fatigue, anxiety, changes in appetite, delayed wound healing, stunted growth and development in children, peripheral neuropathy, impaired glucose tolerance and increased insulin release, subsequently leading to hyperinsulinemia and high plasma free fatty acid concentrations. Studies have documented that chromium deficiency has also led to an increase in hematological parameters, hemoglobin, hematocrit, erythrocytes, leukocytes and mean erythrocyte volume, increased total protein concentration and hyperinsulinemia were observed compared with the group of controls as well. Several trauma and stress may elevate the secretion of glucagon and cortisol hormones, resulting in altered glucose levels, which affects the chromium metabolism. Mild chromium deficiency is a risk factor for metabolic syndrome that may result in heart disease, including hypertriglyceridemia, decreased blood HDL concentrations and hypertension. Let us study the toxicity. Chromium toxicity from food has not been reported, but chromium supplements in high doses by athletes and powerlifters has resulted in some adverse effects with preliminary skin lesions. The hexavalent form of chromium is more toxic than trivalent chromium. An oral administration of 50 mg per gram diet has been found to induce growth depression together with liver and kidney damage in experimental animals. Apart from acute intoxication, chromium toxicity through oral ingestion is apparently not of practical importance for humans. However, workers exposed to chromium compounds used in various industrial processes are known to be carcinogenic and responsible for causing cancer in them. Chromium toxicity is associated mainly with hexavalent chromium, while trivalent chromium is believed to be highly safe mineral. Prasolox in 1999 showed that hexavalent chromium is more soluble than trivalent chromium and at least 5 times as toxic. Lindemann in 1996 gave the safe limit for trivalent chromium to be approximately 1:10,000, and Cohen et al. In 1993 explained that the toxicity of hexavalent chromium compounds is most probably based on an oxidative DNA impairment. Stearns et al. In 1995 explained that genotoxicity may be due to the transistent form of intracellular origin formed by the reduction of hexavalent chromium to trivalent chromium based on D. Flora et al. In 1989 assumption that extracellular reduction of hexavalent chromium to trivalent chromium is regarded as a protective reaction. Chromium, an ultra trace mineral, together with insulin helps in control blood glucose concentrations. It also has a possible role in regulating lipoprotein metabolism. Organic chromium is readily absorbed and quickly passes through the body without being utilized. Inorganic chromium compounds are poorly absorbed regardless of the dose of dietary chromium status. Rates of chromium uptake from the gastrointestinal tract are relatively low and depend on a number of factors including negative valency state, the chemical form, the water solubility of the compound and gastrointestinal transit time. Chromium deficiency is reported to cause impaired glucose tolerance, weight loss and peripheral neuropathy in humans. Thank you, students. Hope that you have understood something about chromium.